Right. So I think we're coming back to another aspect, and we're shortly just going to mention this, and this is gene therapy, another kind of biotechnology. And basically it is also cloning. So we'll just run through this quickly for you. And first of all, how do we do it? And I think, Lorraine, you can come in here.
Goed, geen terapie mense gaan daaroor dat ons 'n normale alleel van die defektiewe geen sit ons in die aangetaste weefsel in. Now this is now to a certain extent what we did with insulin production, we're now doing with an organism. En die navorsing wat in die stadium al gedoen word, onder andere byvoorbeeld op iets soos cystische fibrose. And I'm sure every one of you has heard of cystic fibrosis and might even know somebody with cystic fibrosis. It's a serious genetic disease that affects the lungs and the digestive enzymes, etc. Now what they do is the defective gene, in other words the gene for cystic fibrosis, also has a normal allele. Onthou daar is mos nou mense wat nie die siekte het nie. En daardie gesonde geen wil hulle nou in die selle van die weefsel wat geaffekteer word. Met ander woorde, cystische fibrose affekteer die longe en daarom wil hulle nou gesonde gene in al die selle van die longe inbring. This gene is now supposed to replace the defective gene. They use a vector, and the vector that is used is usually a virus, an adenovirus. That's a weakened form of viruses. So the virus's own genes are removed, replaced with a new gene, and then it is injected into the patient suffering from the disease.
And I hope that as you listen to Lorraine, you can hear this currently is a theory.
Ja, weet jy, hulle experimenteer al daarmee, dit is nog nie suksesvol nie, maar die
hele idee is nog nie
theorie. Ja. Nou goed, nou wat dan nou gebeur is wanneer die virus, ons kyk geweer na ons skyfie, is wanneer die virus nou die sel aanval, want dit is mos wat virusse doen, dan spuit dit die gesonde genetiese materiaal in die selle aan. En dan kan hierdie teikenselle nou die gesonde geen dra. En dan die theorie is, soos Karen ook sê, is dat hierdie gesonde gene nou die probleem gaan oplos. Nou, hulle werk op die oomblik op siektes soos cystische fibrose, soos ek gesê het, selfs hemofilie, spierdystrofie, sickle cell anemie. And all of these, remember, were the diseases that we talked about on Monday. So genetic single gene defects, that's also important. Diseases caused by one faulty gene.
I don't think they can see it so clearly, but they can see that hemophilia and sickle cell anemia is in bold. Those are the two that you have to know for exam purposes. Okay, so I think we have problems too with this, because if they use a virus, you can imagine using a virus as a target to go to that specific organ. How do we know that that virus will end up at that specific organ?
Met ander woorde, die virus moet baie goed geselekteer word. Daar moet baie seker gemaak word dat dit 'n verswakte vorm is, dat dit nie die hele liggaam gaan aantas en ernstige probleme gaan veroorsaak nie, en dat dit net daardie teikenselle sal aanval. The moment the problem, one of the problems is, and I know with this cystic fibrosis research specifically, is that they cannot get the virus to enter all the lung cells and the lung cells only. And when the virus does inject DNA, the takeover process is not always successful.
That's right. And I think we have come to the end of this, of biotechnology, and I hope one day that we'll see a few of them as biotechnologists.
En dit was 'n geweldige klomp werk wat hulle gedoen het. Twee dinge. Gaan kyk na kloning, gaan kyk na insulienproduksie. Goed, en dink 'n bietjie oor, hoe verskil kloning van natuurlike voortplanting? Wanneer ons voortplanting doen, gaan ons vir julle daaroor 'n vraagie vra. Totsiens, sterkte vir die res van die leerwerk vir julle.